Good. Welcome friends, grow with biology. Today we'll study the topic modes of replication and Meselson Stahl Expt. What is replication? Replication is duplication of genetic material. There are three modes which are proposed. They are conservative, semi-conservative and dispersive. Now we will look about all these three modes in detail. First one is conservative. In conservative, we in this diagram we can find out that first diagram which is drawn is in blue color, which is representing the parental generation. And after replication, one is as it is as that of parent and one is comparatively new. One is blue color, same to the parent, and one is comparatively new, that was in green color. So the newly synthesized strand, which is represented in green color, paired upon itself, and there are two separate DNA. One is exactly similar to that of parental and one is exactly the complementary strand which are synthesized on the DNA. Now when we talk about second generation, again the parental strand is remain as it is, and the one the green color which is formed in first generation will again remain as it is, and to both this there will be formation of a complementary DNA that is again green in color. This is conservative mode of replication. Now when we talk about semi-conservative, the parental strand is blue in color. The first generation, the green one is the complementary strand which is um, formed or joined by to the parental strand. So in semi-conservative, after replication, we find that one is parental DNA, another is newly synthesized DNA. In the next generation, that is the second generation, again we find that onto the blue color strand there is synthesis of the green color strand, and onto the green color strand there is again synthesis of green color strand. This is semi-conservative, means one strand will be of parental and another strand will be of the complementary strand. When we talk about dispersive, it is a patchwork of both parental and the newly synthesized strand. So parental is N15 or blue color strand. Now the new strand which is synthesized is green color. So some part is of blue, some part is of green. So it is a patchwork of the parental and the complementary strand. Now Meselson and Stahl did an experiment to prove that what is the exact type of mode of replication followed. Now what did Meselson and Stahl do? Meselson and Stahl has taken the microorganisms and grown it for 14 generation in a N15 medium. So all the nitrogen which is present in these DNA were N15, that is an radioisotopic one or heavier nitrogen. Now they have grown it for one generation in the normal nitrogen, that is N14. Now he find that the density of this DNA which after replication is somewhat lighter than that was what was obtained in the parental generation. Then again in the second generation he grew it in N14, and he found that the parent when we compared with the second generation and first generation, he found that one strand or one position of the band is similar to that of first generation, and another band is still lighter than the previous generation. So in the parental generation, he found the heaviest band. In first generation, they were somewhat intermediate lighter, and in the second generation, he obtained two bands, and one is similar to the first generation, and second generation he has obtained another lighter band. Now these three modes, if we see the conservative mode, one is parental, that is N15. The first generation there is two DNA, one is N15, another is N14, freshly or newly paired, and in the second generation again one is N1515 and rest are N14. Now if this is the mode of replication, then we will find that in first generation there would have two bands. One is N1515, another is N1414. And in second generation also we should have again the similar density bands, which Meselson and Stahl does not obtain. Now, if the DNA has been replicated by semi-conservative mode, then one of the strand in the parental is N1515, and the next generation, the first generation, there is one N15 with N14. So the density gradient centrifugation shows an intermediate or lighter band, that is N14 and N15. In second generation again there is intermediate N14 and N15 along with the newly synthesized N14 N15. In dispersive again it was patchwork. So whenever it was in this mode, then the density will always come to N14 and N15, a combination of N14 and N15. So if when we compare all this with the Meselson and Stahl's experiment, we see that conservative mode has in first generation and second generation it should have two bands and in the exact location. In dispersive there is only one density band, that is a combination of 14 and 15. Whereas in semi-conservative, in first generation there is only one band, that is combination and 14 and 15. Whereas in second generation it has N14 and N15. So this only out of these three, semi-conservative mode of replication only matches with Meselson and Stahl's experiment, which proves that DNA replicates by semi-conservative mode. Thank you.